Welcome to SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode 22. My name is Scott Okuno. I'm a medical oncologist at Mayo Clinic and Chief Medical Officer of SARC. At SARC Talk, we interview interesting people who are connected to sarcoma where we can share and spread the knowledge of sarcoma. Today, our guest on SARC Talk is Dr. Seth Pollack, professor with tenure at Northwestern School of Medicine Division of Hematology and Oncology, and a named professor of the Stephen T. Rosen Professor of Cancer Biology. He did his undergraduate at Haverford College and did his post-baccalaureate pre-med program at Goucher College. He did his medical school at George Washington University as well as his internal medicine training. He then went to the University of Washington, the Fred Hutchinson Cancer Research Center, and did his oncology fellowship. He was on staff at Hutch as well as University of Washington in the Division of Oncology, and then in 2021 came to Northwestern University. Please welcome Dr. Pollack.
Thanks so much, Scott. It's great. It's great to be here.
Yeah, well, thank you. We've been trying to connect here along the way, and our paths on various other venues have met quite a bit. So this is great that we have a little bit of time set aside so we can hear a little bit more. We love to hear stories of how people got interested in sarcoma because some of the stories are not as simple as you think. And more importantly, from your standpoint, if you can share with our audience, a little bit about how you got interested in sarcoma and how you got interested in immunotherapy aspect, because I know that's the main thrust that you were doing.
Sure. Yeah. So, when I started fellowship, I had no idea that I was interested in sarcoma, right? These are rare cancers, and I don't know that I saw a sarcoma patient when I was in residency, to be honest. But I was interested in immunotherapy. This is before the modern era of immunotherapy, certainly before checkpoint inhibitors. Um, I At the time, and this isn't that long ago, right? It's not that long ago, but um, bone marrow transplant seemed like it was still sort of a new thing, and that there might, there were people who were doing non-myeloablative transplants, which was new, and there were going to be all sorts of different ways to manipulate it. And I thought, maybe I'll do. I like the immunology of it, and the Hutch had a lot of great researchers for bone marrow transplant. So I was like, I'm going to go there. I'm going to become a bone marrow transplanter. And as I was doing that, I started to learn about what Steve Rosenberg was doing at the NCI using tumor infiltrating lymphocytes. And I was really interested in that. That sort of was related to the types of things I was interested in bone marrow transplant, that you're taking T cells outside the body and you're growing them into large numbers and infusing them. That's exactly what you do with tumor infiltrating lymphocytes. Just for lay people listening to this, that's when you take a chunk of tumor, somebody's tumor, you grow it in the lab, and the T cells grow, and then infuse it back to people. And they saw amazing responses in some melanoma patients, and now there's an FDA approved TIL product for melanoma patients. And I thought, as I kind of got into fellowship, I was like, oh, maybe that's the kind of thing I want to do. And there was a researcher at the Hutch who was doing a similar sort of thing, but what he was doing is he was taking patients' blood and trying to find the cancer-specific T cells in the patient's blood. And I had a project where we were doing that with trying to find T cells that were specific for a protein called NY-ESO-1. And we would do them actually for a bunch of related proteins, including MAGE4, which is now very important. And I was very interested in trying to take this approach and treat patients with it. And I thought that this is before before Adaptimmune and Telsera, and there were people doing gene modified T cell approaches, but not the big companies that are now important for synovial sarcoma. And we thought, well, synovial sarcoma would be the perfect disease to try and explore this technology with. And we looked for other sarcomas too, and we actually found that another sarcoma type, myxoid round cell liposarcoma, also frequently expressed these proteins. And I had a trial in synovial sarcoma and myxoid round cell liposarcoma of these T cells that we picked out of the patient's blood and grew out to large numbers. And we saw a little bit of response, but it wasn't quite enough to make a big impact on the field, but it was definitely enough that we learned some important things. I think it shaped the direction of some of the research going on with that. I mean, we found myxoid round cell liposarcoma frequently expresses NY-ESO-1, so we did learn things. Um, but but for me personally, it got me to know sarcoma. It got me to know the sarcoma patients, and we had the great Robin Jones from the Royal Marsden was at Seattle at the time, so he became a clinical mentor to me when I was in the last stages of fellowship, and that's how I got into sarcoma.
Wow. So kind of got there a little bit by immunotherapy and by certain histologies, and then you had a clinical mentor out there. Quite interesting. We're going to have a podcast with Dr. Sandra D'Angelo too, talking about the Spearhead trial, and we'll pick her brain too, and that will be coming up. So that's quite interesting. And from our standpoint, you've done some different things over at the Hutch, but you've also been involved a lot in cooperative group studies. You've been involved quite extensively with ECOG Akron, the Alliance Experimental Therapeutic Committee. You've been on the SARC's Discovery Translational Committee, multiple advisory boards, guideline committees for NCCN. These have all kind of helped shape you in your thinking of sarcoma and how we should approach sarcoma. So we're here to kind of pick your brain as how have these relationships and collaborations helped shape how you think about sarcoma and how you're thinking in the future.
Yeah, thanks. Well, what I would say is that each one of these things has its own story in my life, right? So SARC, when I was finishing fellowship, this is a really critical time for young physician scientists, right? Because when you're finishing fellowship, all of a sudden you can go out and generate a lot of revenue for your institution, right? By seeing a lot of patients, and but to be to be honest, when you're at that stage of your career, you don't really have enough skills and accomplishments to be able to justify the NIH giving you like a big grant that would also justify your value to the institution, right? So you really need support during that time, and really you need the way you do that is through some of these career development awards that are really just so important for young investigators. And I got the SARC Career Development Award, and that was another one of the things that solidified my direction into sarcoma. And after that, I had uh, my relationship with SARC became very important to me because of that. So I've been very involved in SARC ever since then. When I was at the Hutch, I have to say we were involved with SARC, but some of these other cooperative groups um, I didn't have a relationship with. So the Hutch is now involved in SWOG, but SWOG at the time didn't have a good strong sarcoma group. Now they do. So I really wasn't that involved with some of these organizations, and I really wished I had been, because I think some people, those groups can be really helpful for supporting young investigators and giving them projects and trials that they can um, take along and really help them advance their career at like a critical time. Um, One thing that I did do is, and I think this is on our list for things to talk about later, but we could just talk about it now. There was a cooperative group that's kind of closed down now called the Cancer Immunotherapy Trials Network, CITN, and and with CITN, I sort of learned that these groups can be really helpful for supporting trials in sarcoma, when you have a new idea and there's not a really clear, sometimes clinical trials can be supported by pharma. If pharma has a great new product that they think is going to make a lot of money, but sometimes you just have a great idea and there's not anybody who has the potential to benefit financially. So we had from my work doing these T cells that I was telling you about before, where we cultured the T cells out from the blood of patients and we infused them into patients, I got very frustrated that I didn't see patients respond as well as I thought they should. And because of that, I went and I looked at a lot of synovial sarcoma patient tumors, and we did RNA sequencing. And one thing that we found is that the sarcoma tumors, the synovial sarcoma and myxoid round cell liposarcoma tumors, really had very low levels of T cells getting into the tumor, and they had very low levels of a protein called MHC. That's one of the that's a very important protein for T cells to be able to see the tumor. And we knew from the very old immunology studies that a drug called interferon gamma could increase the levels of MHC and increase the T cell infiltration into the tumors. And I did a study related to our other studies where I said, okay, well, let's try some of this interferon gamma on our synovial sarcoma patients and myxoid round cell liposarcoma patients. And maybe by just increasing the T cells into the tumor and increasing the MHC into the tumor, we can induce a response. And so we did that in 8 patients, and we did biopsies for every single patient. And we saw that indeed we were able to increase the MHC and T cell infiltration. Um, but um, but but we didn't see responses. So we had a few different ways. Why isn't this working? Well, one thing that we saw is that PD-L1 expression increased, especially in the myeloid cells for these tumors. So PD-L1 is the famous checkpoint inhibitor that in the checkpoint that slows down T cells. It puts the brakes on T cells. And now there's lots of drugs like nivolumab, pembrolizumab, and a bunch of others that can block the PD-1 and PD-L1 interaction. So we had an idea. Well, let's combine pembrolizumab with this interferon gamma, but it was difficult to get the interferon gamma for this new study. But the cooperative group, Cancer Immunotherapy Trials Network, or CITN at the time, was able to help us have a trial that was at several sites around the country where we combined pembrolizumab and interferon gamma. And unfortunately, that trial did not work well. The the patients did not respond. It was a small trial. We're still learning why, but that was a trial that really helped us sort out how interferon gamma and pembrolizumab might work together for these patients. So that's one experience that I've had with the cooperative groups. I'll say another thing. I think that it's important to know that there's a lot we can do without some of these big groups. We don't have to have every time there's a study, it be in one of these cooperative groups, right? There's a lot of really great trials, even in rare tumors like sarcoma, that can be done as single site trials. And that's important because a lot of the times the companies, if you're a company and you have a drug and you want to try it out in sarcoma, you want to test it fast and you want to test it cheaply. And the fastest and cheapest way to test out your drug is always going to be if you can do it at a single site. And that's how we tested our combination of doxorubicin and pembrolizumab, right? So as part of all this other work that I was doing, this is in the very early days of PD-1 and PD-L1, when we were just even before FDA approval, when we were just starting to see the great signals of activity. We had a collaboration with Merck where we, partly based on the work that I've been doing in synovial sarcoma, where we tested a whole bunch of sarcomas for PD-L1 expression, for T cell infiltration. We looked at really fancy stuff, like we looked at the TCR repertoire. So we looked at all kinds of stuff, and and out of that work came our trial of doxorubicin and pembrolizumab, and that was a trial where we tested doxorubicin, which is standard frontline treatment for most patients with metastatic sarcoma, and we just simply added in pembrolizumab, and we were able to do that as a single site trial, and we got it done relatively quickly, and we had some good data from it. We showed some impressive we showed a good response rate with impressive progression-free survival, particularly in patients with undifferentiated pleomorphic sarcoma and with dedifferentiated liposarcoma as well. And then another cancer center at the Levine Cancer Center did a similar study, and they found that all of their patients with undifferentiated pleomorphic sarcoma responded.
I just want to go back to something that you said, and I know you quite benefit from a career development award through SARC, and that solidified your I'm in, I need to do it. SARC has continued to work in to revitalize the CDA because I think it really launched many of our sarcoma thought leaders and productivity and made people aware that sarcoma is a field that you can actually spend a career in. But here we are looking at you down the road and seeing all these groups that you've worked on, worked with, and you're starting to pick and choose where each of these strengths are. You talked about the CITN network. They had some strengths. You leveraged them. You've been at the Hutch. You've been at Northwestern. We'll talk a little bit more about that. There are values of doing stuff there. You and I belong to the Midwest Sarcoma Trials Partnership. So we know there's value in cooperative groups. You are heavily involved with SARC, which is a cooperative group as well. So part of these has strength and values, but I would ask you, if you're a young, new sarcoma researcher interested, they're not going to be able to navigate years and years that you've taken to get to this point. What advice will you have for somebody who is just starting out? Are you going to join every single cooperative group? Because it's mind-boggling. It
No, no, no,
is.
no, no. So I don't think for young investigators, I shouldn't say this, because there are a few people that have had trials through the cooperative groups, and that's really made their career when they're a young investigator, for sure. The to do that, you kind of have to have a mentor that's got a trial all ready for you with a bow, that can just kind of hand it to you and you sort of take it over, right? I think for most, we're going to talk in a little bit about my big trial with ECOG Akron, right? The phase 3 trial that's just opening right now. That took years to get off the ground, right? And that was based on the trial that I was just talking about. So I think for most of the time, for a young investigator, it's good to participate in this sort of thing a little bit, because it's a great way to make connections. It's a great networking opportunity. But I think for for somebody just getting started, I think it's good to try and do some retrospective sorts of studies, right? Because you're going to be able to do that, and you're going to be able to get it published, and it's going to give you a record that you can then try and build off of when you go to industry and try and get trials and stuff like that. You can be able to say, hey, look, I really know about this patient population because I've looked at this population carefully and I've proven it by publishing my work. I think, as I was saying before, like the doxorubicin pembrolizumab trial that I did when I was at the Hutch, that was a trial that I was able to basically get off the ground with help from my mentors, right? But it didn't take me years and years to get it going, right? Cooperative group trials take years because you have to, there's so many different committees you have to go through. So I think that they're very helpful in terms of networking. If I was a young investigator, or if I'm counseling a young investigator, I'd say pick one, go to all the meetings, including the in-person meetings, so that you get to know the people and like really know the people. Go to the SARC meeting because it's so easy to go to. It's like right before the CTOS or the ASCO meeting, and try and actually introduce yourself to the people at the meeting, get to know them, because I think the real value in those organizations is for young investigators is the networking opportunities.
No, that's very, very helpful. And I'm going to go back to the discussion you had about it is okay to do single institution studies. And that's what you did. You know, the Levine Cancer did a group, and then maybe share how those two concepts were used to help develop the long journey
Yeah,
that you finally, and you and I talked about it, is say, whoa, it's finally here. So it's still a relevant study, but oh my gosh, if we wait too much longer, it might not be, but go ahead.
the doxorubicin, dox pembro. Yeah, I mean it's it so um, it was a long journey to get that trial open. So we had our single institution trial, which was great. I mean, it was great. I know that there's patients out there that have been treated with doxorubicin pembrolizumab just based on this phase 1/2 data. That's not what I'm recommending, right? I'm recommending people participate in the trial because we really don't know whether this is an improvement on over standard of care. We had great results, but it's not uncommon for you to see great results in a small pilot trial, right? Because your patients sometimes the patients that go on these trials are the patients that are in the best shape. They're fit. Sometimes these are the patients that are shopping around for, oh, I want to go to the center that has a new exciting trial. Um, Sometimes there's like some sort of investigator bias that's subconscious where they say, oh, they don't even know why they're recommending this patient for this trial, but or they're kind of like saying to the patient, I really think you should do this trial. But it may be that there's something about them that they know that they'll fit, but when you try and take it broader, you can't put your finger on what that thing is. So you really need a randomized trial to show, um, unless it's a total home run, right? But like the T cell therapy that we were talking about before, right? Which isn't a total home run, but that's a new treatment where people's tumors really shrink, and there probably is a small percentage of patients where it's a true home run.
Yeah, I'll just so people are listening, it's a Spearhead 1 trial. So just in case you want to Google it, it's for synovial myxoid liposarcoma. We'll talk with Dr. D'Angelo about that a little bit more next
Yeah,
month.
yeah. I didn't mean to get too deep into it, but so like the trial with ECOG Akron, what we said is okay. We want to really test this concept of combining doxorubicin and pembrolizumab. Um, so the only way you can do it is to have a randomized trial, and for that you really need a cooperative group. That's one of the situations where I think you can only do a trial if you have a cooperative group, is when you're trying to influence the standard of care. You need a multi-institutional study, and with these sorts of drugs that aren't Merck has a lot on its plate, and I don't think that they would have been willing to support the study just as a Merck sponsored trial. So we really they are contributing a lot to support the trial through the cooperative group, but the cooperative group is really sponsoring the trial and allowing it to be this big multi-center trial. So this is a trial that's going to be held at that is being held at institutions all across the country. It's really for patients with undifferentiated pleomorphic sarcoma who have not been on anthracycline before. That means they haven't received either AIM or MAI or MAID or any of the other combinations that include doxorubicin. Um, these are for patients with unresectable or metastatic disease, and their patients are getting either randomized to receive doxorubicin upfront or doxorubicin with pembrolizumab. And then if they're on doxorubicin and they eventually progress, they can get pembrolizumab alone. So this was originally proposed as a phase 2 trial, and it was also going to include dedifferentiated liposarcoma, which we also thought would benefit from the combination. We went through multiple iterations of the trial. We had to drop dedifferentiated liposarcoma at one point. We got the trial almost ready to open as a large randomized phase 2 trial, and we had to stop. The FDA got involved and said, hey, if you're doing this trial, we really think that this should be done as a phase 3 trial. So we had to go back, go through all the committees again as a phase 3 trial. And now it's finally open, and I think it's accruing really well. I mean, it hasn't even been open for 4 or 5 months, and sites are still opening, and it's accruing. So I'm excited about the possibility. I think it's going to open in Canada. We're looking for ways to open it in other countries as well. And I think we're even looking at possibilities, other possibilities to try and expand it. So it's something I'm really excited about. But talking about cooperative groups, this is one of the things that I think the cooperative group offers. I do want to say one other thing about the cooperative groups, if that's okay, Scott. Yeah. So there's two real great, cooperative groups can do a lot of stuff, but there's two things that I think the cooperative groups can do that single sites really can't do. And one is a trial like this where you're really trying to push forward the standard of care, and you need a lot of patients, right? So a cooperative group study can really help you with that. The other one that I think a cooperative group study can help with is when you're looking at a rare subtype. And that the rare subtype is something that a single institution, you're not going to be able to accrue enough patients, right? And I'm thinking of the ETCTN trial of alveolar soft part sarcoma, where they really definitively showed that a PDL1 inhibitor could improve outcomes for alveolar soft part sarcoma. And that one, I think there's a danger, because I think right now there's a little bit of a pushback against some of these cooperative group trials that focus on the ultra rare sarcoma subtypes. And I think that one thing that I hope the sarcoma community will do is to maybe push back a little bit and say, no, these trials in the ultra rare sarcoma subtypes are important, because we do have these in, we had the tezemetostat trial. Some of the companies will do these, but for trials like the one in alveolar soft part sarcoma, we're really going to need support to get those done.
Absolutely. Just to follow up a little bit, Seth, your perspective is very helpful for all of us to kind of reframe our thinking of the value of cooperative groups. And I think one of the strengths of SARC is that these are all sarcoma centers that do this. Now, cooperative groups, if you're part of the cooperative groups, even if you don't see a lot of sarcomas, you can activate this study. Most oncologists know how to use doxorubicin, and most know how to use pembro. So if you need to answer a question very quickly, definitively, I think the cooperative group, despite the importance of all these committees and reviews that Seth had to go through, is a strength. And I think you'll get the answer quicker than if you did it. You'll never get it at a single institution. And only sarcoma centers, we don't have the infrastructure and finances to do that. That's why the cooperative groups are there. Maybe I'm speaking to the choir, Seth, but your thoughts. Yeah.
Yeah. So SARC is kind of like, I feel like it's different than some of these NCI supported cooperative groups, right? It's a network of sarcoma centers, which it's actually a great way if you're a company and you want to get done a trial in sarcoma. I think working with SARC is like probably the best way to do it, right? Because you can just go, you've got like a sort of like a direct line right to the all the sarcoma centers that are going to see the most sarcoma patients, and I think it's a great way to do it in a rare patient population. um, and it's a great way to do it in a lot of centers because I don't know about them. I don't know the numbers in terms of if it's a majority, but a big percentage of the sarcoma patients are seen at sarcoma centers, more than say breast cancer, lung cancer, colon cancer, where I think a lot of the sarcoma patients have come to recognize that they're going to get better care at a place that sees a lot of sarcoma patients. I think that a lot of the sarcoma patients are younger and they're a little bit more motivated to go travel a little bit farther to the places that are going to be expert. Of course, the the thing that SARC doesn't have that the NCI supported cooperative groups have is the NCI support. So it's it's there's pros and cons with any mechanism.
100%. We would love to have more support, and anybody listening, we'll be happy to chat. I'd like to go back to your decision in 2021, or probably beforehand, to leave the Hutch and to come to Northwestern because you had a vision when you came to Northwestern, and I know that the practice was there when Mark Golnick was there before he left, and then you're building a practice there, and that will tie into your IITs that you have also at Northwestern. But kind of what was your plan when you came to Northwestern and how you're building that out? Because I do know you had some good colleagues that you're building up as well.
Yeah, well, I had a great education at the Hutch, and I was able to do a lot of these trials like I'm talking about. But um, I felt to take it to the next level, I had to really um, I had to basically come into a new situation where I was starting from day one with this vision that I was telling you about, and or that you're just talking about, and with a when you're recruited to someplace, right, and you have a vision, it's basically like the leadership is saying, okay, we're hiring you and your vision for building it, right? Um, Everyone at the Hutch was always supportive of what I was doing, but I kind of grew into it, right? So I was always trying to um, push my vision into it. So to build this up here, I think the first thing that I had to do was to have another sarcoma person, right? Because Northwestern is actually a big institution, and we're the biggest sarcoma center in Chicago. Where we actually see about half the sarcoma patients in Chicago at Northwestern downtown. So, and I can't be running a lab and having trials and seeing all the patients here. So we had to develop a new sarcoma specialist, and that's Pedro Vivieros. And he is fantastic. He was here at Northwestern doing a phase 1 clinical trial fellowship when I came on, and he started doing clinics with me. He really liked sarcoma, and I think he was really excited because we right away had a lot of clinical trials going in on sarcoma. He's very interested in clinical research, and now he's seeing most of the sarcoma patients here at Northwestern. So that's something I'm one of the things I'm most proud of in terms of sarcoma here, and we're developing another sarcoma physician as well, actually a Mayo Clinic grad, Fahad Faruqi. He is right now he's based at Northwestern's. We have a hospital in Warrenville. It's called CDH. It's it's not the main hospital downtown, but it's in the western suburbs of Chicago. And we've been sending him. So most of the patients that come from the western suburbs, he's been the one that we send them back to. So he's been able to develop sort of a specialty practice in the West. And he comes to all of our sarcoma research meetings. And he's even though not all of his patients are sarcoma, he's really developing an expertise. I think now probably about half of his patients are sarcoma. So he's a knowledgeable person. He comes to all our tumor boards, and I think we're trying to make it so that he can come see patients downtown for 1 day a week. So that's something that's in the works. And there's a doc in the north that we're trying to basically replicate this success with, Bindia Patel, who is building up a sarcoma practice there. So yeah, I mean, we're really building things up. We have some trials that we've started, and we've got a few more that we're working on. So yeah, I'm so happy with how things have been going at Northwestern.
For us that have lived in Chicago, going downtown is not a simple feat. So just to let you know, so having good sarcoma expertise that is overseen by the sarcoma team that you guys have out in the suburbs is outstanding, and it's very exciting because it will allow you to feel very comfortable of extending the care that sarcoma is at Northwestern to reliable and engaged partners. But that also comes back to your vision to have IITs at Northwestern. And you have some, and maybe if you're comfortable sharing, what are your IITs that you have ongoing and preluding a little bit of what's going on at CTOS and some of those studies. So maybe some insight from, are you willing to share?
Well, so I've had a bunch of IITs in the past, right? And right now we have just the one IIT that's open, which is this XL092 study. So this is a trial. It's a single arm phase 2 study of a new drug called zanzalizumab, and zanzalizumab is made by a company called Exelixis. For those of you who know about sarcoma drugs, it's a tyrosine kinase inhibitor, which means it's in the same class as pazopanib and cabozantinib and regorafenib. But we think that, or there's reason to think that it may be more effective and it may be better tolerated than some of the other drugs in the class. So this is a phase 2 trial in leiomyosarcoma right now that's open. And we've already started treating patients. And so far, I do think that it's well tolerated. It's too soon for me to comment on the efficacy. I've had a number of other trials, IITs in the past. We've talked in the past about our TLR4 trial. This was a trial where um, I'm sorry. We do have one other IIT I should mention. It's there's it's kind of related to the TLR4 trial though. So when I was at the Hutch, I had an IIT where we TLR4 means toll-like receptor 4. TLR is these TLRs. They're some of the oldest ways that the immune system knows that there is a problem, right? So it's basically there are molecules on bacteria and fungi that the immune system can directly recognize, right? It doesn't need to have an antibody. It doesn't need to see a virus inside the cell. It sees this and it knows there's a problem. And what we did is we used this TLR4 agonist, and this is basically it's this particular TLR4 agonist is part of is basically copied from a bacterial cell wall. And we would inject it right into the patient's tumors and give it along with radiation. And we saw very good responses. We saw some tumors, we saw one tumor at least disappear completely. A number of other patients had very deep, durable responses. And but the biggest thing is that was really interesting, that made us excited for the future, was that we saw these big T cell infiltrations with T cell responses that seem to be recognizing the cancer for a variety of reasons that I won't go into. And this made me think that um, it's important to get the immune system's attention to something specific when you're giving radiation. And there's a radiation oncologist here at Northwestern that's been working a lot on this new technique that's becoming fashionable in radiation oncologists in oncology called grid technique. Grid is a technique of radiation where you give, instead of just radiating the whole tumor with sort of a certain dose, you give beams of like super high dose radiation. And basically what you get is they'll sometimes call it a lattice, because uh, you get like kind of a wave of high doses of radiation and lower doses of radiation. And that can, what that does is it gives it's actually a really potent way to give radiation. But what I was interested in is that it causes release of what are called damage associated molecular patterns. So there are these specific proteins that the immune system will recognize when um, when cells are killed quickly. And these excite the immune system in very similar ways to the TLR4. So we have a trial right now where patients with retroperitoneal sarcomas can receive proton grid, and this is another IIT. I think about it as immunotherapy IIT because we're collecting a lot of data from the patients that is immunologic data, because that's something that I'm interested in. But it's not really an immunotherapy trial in the sense that we're not using a new immunologically targeted agent. It's this proton grid technique. And what's unique about this is, because there are a lot of people around testing grid, we're using protons to deliver the grid treatment. And the one thing that's nice about protons is you can give them very accurately, which, other people I think have been scared to use this sort of grid technique in the retroperitoneal area, where you could accidentally give a high dose of radiation to the bowels or to the kidneys or something else that's in the abdomen, right? But the proton grid lets you be very confident that you're delivering all the radiation to the tumor. So that's the other IIT that we have open now. We do have a number of other projects that we're trying to develop.
Well, as you bring up that point, is that sarcoma management is not just medical oncologist. It's the radiation oncologist, it's the imaging, it's the pathologist. And what your team is doing in Northwestern is building upon those with correlative studies. And I think that is the exciting piece that you are building out. What I'm starting to hear is the excitement that you have, the vision. There's a lot of research ideas. There's a lot of ways for us to communicate. And in the past, we used to spend time together in a room batting it out, as we would say. A lot of our meetings are like this, is more virtual and that. And I think there's value in putting together time to discuss things. And where do you see us in the sarcoma community having the time to stop and have the in-depth discussions that you are sharing with us today? Because we get sound bites. That's all we get. Seth, it's just very hard.
It's so hard. It really is. It's hard. It's hard because I think for everybody, um, it's the things you're saying, but there's another aspect to it where I think physicians, other healthcare practitioners, scientists, there's more and more that we're being asked to do, and people are having more and more demands of their time. And the it used to be that if you were in academics, people had a real reverence for the academic mission. People, maybe there was a, people talk about ivory tower now, like just in this, like only a negative light, that you're kind of like off in your head, but there was something really valuable to it, that you have time for reading, that you have time to think about things. And I think it's very hard to do that right now. I think it's hard for me to keep up with what's happening with all the, publications that are happening in sarcoma, right? I don't know how people do it in common cancers where there's even more literature. But I can't, it takes a lot of effort to keep up, on the cutting edge with what's happening in sarcoma. And with immunotherapy, I mean, it's even more, right? So to stay up with that, I have to really. It takes a lot of effort just to manage my time, to be honest, to be able to do that, and then to add in what you're talking about, which is to be collaborative about it. It really takes an effort. So I think the place to start is with some of these meetings that we have with the SARC meeting. The SARC meeting is so valuable for that. CTOS is also a great meeting for discussing issues related to sarcoma and meeting with people. I have great discussions at ASCO. For a while, I used to mourn that ASCO got so huge that really you can't see everything anymore. But I mean, you could never really see everything. But I mean, now it's even, I just have to focus on the sarcoma meetings that I'm interested in at ASCO. But what that's done is that's freed me up the rest of the time to just be meeting with people and talking about different things. Um, and then I think for me, there's an issue. You can have too many collaborations, right? I think it's where you're talking to a lot of people about a lot of different things and nothing's actually getting done. And I think that I have to make an effort to focus on the quality versus the quantity of collaborations, right? So I have like a handful of collaborators that I know that I work really well with. We meet regularly and write grants together so that there's like an actual product that comes out of it. Hopefully we'll get funding from it. And that's an important aspect of collaboration.
Well, Seth, I know our time is coming up here to the close pretty soon, but you've shared a lot of insight on career development, on what cooperative groups, single institution, development of your career over time. And the last piece that I just started thinking about is being very intentional of your time, because people are putting more and more on our plate, as you just said, just because what we do, but you're being very selective what you put on your plate. And I think that's the part that it's the aha moment. Like, yeah, so that's interesting because yeah, we love to be everywhere, but if we have control, we will decide what we go on our plate. So interesting. So as we end here, we just like to get your thoughts. And if you had a billboard, and Chicago has a lot of billboards, and those are listening to SARC Talk here, what would the billboard be for your message for sarcoma and immunotherapy in the next 5 years? And you had a billboard, so it's kind of an elevator speech. What do you see in the next 5 years on that billboard? And we're going to hold you to it because when we interview you again or have you on SARC Talk, we'll do it.
If it's a billboard, you can just fit a few words on a billboard that I think, right? So I would say just keep pushing forward, right? Because there's going to be setbacks, right? But the only way that you can move forward is if you just keep putting one foot in front of the other, and that's how we make progress.
Oh, well, that's what I'm going to remember. Keep moving forward, okay? As they say. Seth, any other comments or things that you might have for our audience?
No, this is great, Scott. Thanks so much for having me.
No, we appreciate it, Seth, and thanks for sharing your time and insight with us. And we look forward to meeting next month at CTOS. So this is
Yeah, there
good.
you go. I'll see you next month.
So thanks everybody for listening. Looking forward to our next discussion with Dr. Sandra D'Angelo as well. in December. But it's been great, Seth, spending some time with you. So thank you.
Thank you for listening. Be sure to subscribe so you never miss an episode of SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or for questions, please email us at sarctalk@sarctrials.org. The SARC Talk series is made possible due to funding from Boehringer Ingelheim. Together, we can find a cure for all forms of sarcoma.